Cancer is a leading cause of death around the world, and its incidence continues to rise. Each year, 12.7 million people discover that they have cancer, and 7.6 million die from the disease. However, evidence shows that 30 to 40% of these deaths can be prevented, and one third can be cured through early diagnosis and treatment. For breast cancer, the survival rate, if detected early, is 98%. Therefore, today we will talk about a device which helps detect cancer at a very early stage. These are biosensors which work on the principle of surface plasmon resonance, that is SPR.
So, what exactly are biosensors?
Biosensors are devices that are designed to detect a specific biological analyte by essentially converting a biological entity, that is protein, DNA, or RNA, into an electrical signal that can be detected and analyzed. In terms of cancer, the analyte being detected is a tumor biomarker. By measuring levels of certain proteins expressed or secreted by tumor cells, biosensors can detect whether a tumor is present, whether it is benign or cancerous. Optical biosensors are based on the phenomena of surface plasmon resonance techniques. But what are surface plasmon?
So, let us see what surface plasmons are. By definition, surface plasmons are coherent, delocalized oscillations of electrons that exist at interfaces of two mediums where the real part of the permittivity change is sign. Too complex, so let us understand it in a simpler form. In surface plasmons, there exist evanescent waves on both sides of the interface of two mediums whose permittivity is of opposite signs. Now let us understand what evanescent waves are. When the angle of incidence is greater than the critical angle, then a part of the incident energy is carried by waves called evanescent waves, which are formed in the second medium. One of the ways to create surface plasmons is to have evanescent waves incident on the dielectric metal surface. We know from boundary conditions that the kx of the waves in both the mediums should be equal. Also, for the formation of surface plasmons, we can derive that the kx of the waves in the second medium is a function only of the permittivity of the mediums. So, if we fix the frequency of the incident energy source and vary the angle of incidence, then surface plasmons will be created at a particular angle of incidence, and at that angle, majority of the incident energy would be carried by the surface plasmons into the second medium, and so a dip will be observed in the intensity of the reflected waves at that particular angle. Since this angle is dependent only on the permittivity of the two mediums, we can find out the properties of the second medium if we know this angle. A glass slide coated with a thin gold film creates the sensor surface. For most applications, a dextran matrix covering the gold film acts as a substrate to which molecules can be attached and provides a hydrophilic environment for the interaction. Other matrices can be used to attach specific types of molecules. The specificity of the surface is determined by the nature of the molecules that attach to it. So, one binding partner is attached to the surface of a sensor chip and the other is injected in a continuous flow of solution. Whatever the nature of the molecules involved, we call the attached interacting partner the ligand and the partner in solution the analyte. The device uses the phenomenon of surface plasmon resonance to detect biomolecular interactions as they happen. SPR causes a reduction in the intensity of light reflected at a specific angle from the glass slide at the sensor surface. As molecules bind to the sensor surface, the refractive index close to the surface changes, altering the angle of minimum reflected intensity. The change in SPR angle is proportional to the mass of material bound. The sensor surface, the microfluidic system, and the SPR detection unit work together to measure biomolecular interaction. The result from the detection of change in refractive index is displayed as a sensor graph with the binding response on the y-axis is plotted against time in the x-axis. Since light does not penetrate the sample, analysis can be performed on colored, turbid, or opaque samples. From studying the shape of the sensor graph produced, binding yes or no, specificity, affinity, kinetics, and active binding concentration can be determined.
This is really interesting, man. An excellent application of the concepts we studied at class. Are you aware of the future scope in this field? I'll tell you. One of the ultimate goals of biosensor technology is POC cancer diagnosis, point of care testing or diagnostic testing that is done on site is a field in which biosensors could have a major impact, allowing the patient and medical staff to get results rapidly and easily. POCT allows for faster diagnosis and has potential to significantly reduce costs. Thank you.